Hello, everyone, and welcome to today's webinar titled Enhancing Laboratory Efficiency, Automating Plasmid Protein and Cell Processing. This webinar is a part of the 8th annual event in the Laboratory Automation Virtual Event Series. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by GenScript. For more information about GenScript, please go to www.genscript.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you first to participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You will also notice that the speaker has provided a survey located on the left of your screen. This survey will be available during both the live session and the on-demand period. Now, if you have trouble seeing or hearing the presentation, please click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. All right, now let's get started. I'd like to present today's speaker, Rouba Najjar, Head of U.S. Marketing and Business Development, Products Division. For a complete biography on our speaker, please visit the Presenter tab from the menu on the left of your screen. Rouba, thank you for joining us. You may now begin your presentation.
Thank you, Kaylee, and thank you for the audience for joining me today. As you heard, my name is Rouba Najjar, and I am responsible for the Products Division over at GenScript. So for today's presentation, I've really tried to focus the discussion towards presenting you with a lot of the solutions that are coming from GenScript. And you'll see that I've also included in the presentation some overview information with regards to automation and the laboratory processes in general. So I hope this is informative, and with that, I'll go ahead and get started. So I'm going to start the presentation by just giving you a brief overview of what the main focus of this presentation is going to be. So I'll go ahead and do an introduction about GenScript as well as lab automation in general. And then after that, I'm going to be talking specifically about some of the solutions that are going to be basically targeted towards sample processes. These solutions include plasmid purification, protein and antibody, as well as cell isolation and activation. So GenScript has been around for more than 20 years. We've really developed quite a bit of expertise in different disciplines, and we're a global company. As you can see on the map at the slide, we have a couple different headquarters throughout the globe. Just briefly, a couple milestones for GenScript. We were founded in New Jersey. This is where I'm based as well. So it's really one of the bigger headquarters. We have a secondary headquarter in China, and we have a couple different headquarters throughout the globe. GenScript is publicly traded. We were initially listed on the Hong Kong Exchange in 2015. And we have a couple different divisions, including a Legend Biotech, which is really well known for the cell therapy. We also have our division that is called ProBio, which is a CDMO division. And since then, since our initial launch, we've actually expanded in additional spaces, such as synthetic biology, which is a division that's called BestSign. So as I mentioned earlier, my main focus in this presentation is to kind of provide you also with a background on some of the automation workflows. So one of the main things, of course, is that we're all faced consistently in a fast-paced environment with challenges in regards to optimizing our efficiency. So what I wanted to kind of deliver on this particular slide that you're seeing is that typically when we're looking at sample preparation and research efficiency, Typically, our laboratory professional or a researcher is spending between 20 to 70% of their time just on the sample preparation. In addition to that, a huge section of their budget is dedicated just for sample preparation. On average, it's between 50 to 60%, which of course is extremely cost prohibitive, and basically the research dollar doesn't stretch. Furthermore, this is also going to be extremely prohibitive if you don't have high quality samples and a good purification process that you're implementing. So purification, as well as a high and a crucial setup, is going to be very important for this success. So what you're seeing here is that for a traditional purification method, I broke it down basically by the average time required for different processes. You can see, for example, for a plasmid maxi scale, it typically takes, just for that purification step, between 4 to 8 hours. Another instrumental purification method would be, for example, protein or antibody purification. And that typically takes between 2 to 12 hours. So very, very variable, but also very, very time consuming. There's a couple other examples that I listed out here. I'm not going to go into each one of them for the purpose of time. But overall, the main point here is that basically a lot of these purification or preparation methods are extremely time consuming. This is a quick overview of the evolution of automation in life sciences and biotechnology. Basically, we're looking at from the very early phases of automation, where it was started in the 1950s with a very simple setup of an auto analyzer, all the way to where we are today, where basically we've expanded from an initial setup that is semi-automated to a high throughput setup to where we are today, which is basically complete remote work, or maybe even personalized medicine. Of course, with that said, there's a lot of still challenges and a lot of issues that come up, but overall, this has really improved not only the outcome of research, but also the actual experience from a laboratory perspective and a laboratory user. So another thing to mention here is the impact of automation. I basically plotted a graph to the right-hand side showing basically how laboratory automation has really changed the efficiency in a laboratory setting. So you can see there was a pretty significant growth between the 1970s to the 2000s. Right now, it's becoming a little bit less of a drastic jump, but certainly it is still ongoing. And really, the main things to highlight here is that there's a huge benefit for automation when we're looking at the amount of time that is being spent on laboratory prep. So the estimate for that is up to 70%. From the previous slide that you saw, it's between 20 to 70%. A huge benefit, of course, is the reduction of error. So on average, the automation typically decreases the error rate by more than 50%. Another key benefit would be the cost reduction. So not only is it from a process standpoint, the cost reduction, but also from a manual setup and the time that is being invested whenever we're doing prep. Another key benefit, of course, would be the operational efficiency. So we've really taken processes that might not be very streamlined, might not be very efficient, and we've taken those and automated them. And then, of course, the last key point to mention here is the technological advancements. Whether it's using different methodologies to prep the sample, or maybe even optimizing further with multiple different integrations, automation has been really key in that. And with that, I would like to start going into the details of the products that are offered by GenScript. You can see this is basically an overview slide. This overview slide, I'm not going to go into it right now, but the gist here is that we're going from basically a DNA assembly to plasmid purification, to protein and antibody, as well as the analysis using electrophoresis and Western, all the way to cell isolation and activation. Now I'm going to be talking about our automated maxi-scale purification system. The system is actually very close and dear to me because I personally have had the experience of purifying maxi preps for quite a bit of time. It was an extremely painful process, very tedious, very time consuming. And in some cases it would fail, which made it even worse, of course. So when we're talking about plasmids, it's really important to think about, one, the actual quantity that you need, as well as the quality that is needed. So if you're working with more smaller scale plasmids with basically research level, you're doing maybe cloning, mutagenesis, applications such as that, you're typically working with a lot smaller quantities of DNA. However, the higher up you go, the more stringent the protocol is going to be, and the more quantity, more DNA you're going to want to get out of that prep. So when you get closer to the transfection, when you get closer to viral production, or maybe even DNA vaccines, this is where the amount of DNA you need is going to be significantly more, but also the quality of the plasmid is going to be a lot more challenging for you. So I think that I probably am not presenting anything groundbreaking here, but I did want to mention here that basically, of course, plasmid purification has been around for many, many years. I think the initial identification of a plasmid was in the early 70s, where they actually were able to isolate it. And there's been quite a bit of methods that have been developed to be able to extract that plasmid. Of course, we're all very familiar with the ion exchange column methodology. That's really where a lot of the development has been focused on. But with that, a lot of it, of course, is very, very tedious, error prone, and of course, extremely time consuming. What you're going to see next is basically the solution that GenScript has developed, which is based on the magnetic silica beads, which is what you're seeing on the right hand side. Before I go into the GenScript solution, I did want to briefly touch on the actual plasmid purification workflow. Of course, it starts with basically growing your culture offline. Typically, you're using different methods or different media. I apologize. And of course, once you have your culture grown, the very first step is to go ahead and centrifuge it down, followed by a series of steps within the workflow, such as resuspension, lysis of the bacterial pellet, neutralizing of the bacterial pellet, followed by multiple steps that include binding and washing. And of course, every single step of these is basically coupled with a centrifugation step, which takes an incredible amount of time. And of course, all of this is hands-on time. With that said, what we had come up with is what we call the AmMag Quattro. This system is capable of automating maxi-scale plasmid purification. The system is really modular. So what you're seeing on the image right here is a controller, which is the smaller unit on the right-hand side. And adjacent to it, you're seeing the purification modules. So what you're seeing here is a set of 4 purification modules, and each module is capable of purifying up to 6 samples at a time. So if someone is interested in automating their purification workflow, they can technically start with 1 module. And that one controller, what you're seeing on the right-hand side, as I mentioned earlier, is capable of controlling 1, 2, 3, or 4. And each one is completely independent of one another, which of course is an essential setup when you're working with automation. You have to think of the scalability of your workflow. You have to think of also how you can be flexible in your workflow. So each single one of those modules is completely independent of one another. So for example, if you want to run them at different times, if you want to run them at different protocols, it is not a problem. Another thing to highlight about this solution is that it is actually a complete solution. We have designed the consumables that go hand in hand with this system. And these consumables are basically implemented in such a manner where they're going to be ensuring your success to basically fully automate maxi-scale plasmid purification. The next thing that is coupled with this system is the pre-programmed verified protocols. Those protocols are preloaded into the system, so you don't actually have to do any additional programming unless you choose to. So theoretically, this is an out-of-the-box system. So you can get the system on hand, take it out of the box, get the consumable, and of course your culture, and within that same day, you can actually run the system. So some of the main features of the AmMagQuattro is, as I mentioned earlier, is going to be really this fully automated workflow, all the way from the bacterial pellet to the purified plasmid DNA. The system is high throughput in the sense that if you end up getting a 4-module setup, you can run up to 24 samples at a time. The pipetting accuracy of this liquid handler is a CV of 3%, which is really good. And the culture volume, where you're basically growing your bacterial pellet, is going to be really a wide range between 25 to 250 ml. It really depends on the plasmid you're growing. It depends on the insert. Of course, there's a multitude of different conditions that might impact how robust your culture is. That's not really the limitation. The limitation is actually by the bacterial pellet weight once you centrifuge that culture down. So really the optimal range of the bacterial pellet is going to be 1 to 1.5 grams of bacterial pellet. So this is really the sweet spot of the instrument or the solution. And it typically takes about 2 hours to run the AmMagQuattro. So theoretically, you can run the system 3, maybe even 4 times in a day. So that 24 sample, if you have a 4-module setup, could be double, triple, or quadruple, depending on how many times or what your throughput is. And what you will be getting is actually transfection grade plasmid. So the endotoxin level is going to be less than 0.1 EU per microgram. And the plasmid itself is of high quality. So the super cold ratio is going to be exceeding 80 to 90%. And as I mentioned before, the consumable kit is included as part of the solution. And I have a designated slide that I'll show you in the later deck. And what I do want to highlight here is that the consumable kit is all inclusive. So it's going to basically include everything that is needed to run the system. So you're not going to have to basically supply your own tips or tubes or anything in that manner. It does include all of those key components. Lastly, the system also includes a very easy, user-friendly interface. So you can certainly use the protocols that we have supplied for you, but if you choose to either develop your own or optimize the protocols that we have supplied, you can certainly do so using the interface. So to take a few minutes here to talk about the all-inclusive consumable kit. So what you're seeing on the right-hand side is basically all the key components that go into the kit, the actual tips, the final elution tube, which is the green cap tube that you're seeing on the left-hand corner, the cartridge that contains all the buffers, the magnetic beads, as well as the elution, the sample tubes, which is what you're seeing as labeled as C and D, as well as the waste container. So basically all-inclusive. And the position on the right-hand side is showing you the different compartments of where each consumable goes. I'm not going to go into that because of the time limitation, but of course, I'm happy to answer any questions later on. So another key feature here I want to mention, which I had touched on earlier when I was referring to the benefits of automation as a whole, and which is the cost efficiency that the system is going to provide you with. The actual consumable kit is actually less than the market average of the manual kits. So if you're currently using a manual kit that is commercially available, whether it's one of the more popular names, I'm not going to mention anybody, but you're going to basically see a very nice cost efficiency if you end up choosing to go with this solution, between 20 or even 30% of the kit price. Of the discount from the kit price. Another thing to highlight here is that basically the commercially available kits do not include the tips, the tubes, the waste, all of those key components. So in addition to the cost efficiency, we're also adding additional components that typically you have to also supplement with your kit. So here I have prepared kind of a quick overview of the plasmid purification, the workflow that is being done on the AmMagQuattro. So this is not what the user would be doing. I just wanted to quickly provide an overview of how it works, basically. So the very first step is basically to centrifuge your bacterial culture down. In the central section of the slide, where basically you're seeing a very wide box, is what basically would be fully automated on the AmMagQuattro. So the system is going to take your bacterial pellet, it's going to go ahead and resuspend it, perform the lysis, the neutralization, all the way to the elution. At the very final step, which you probably remember, I showed that single green tube. That single green tube contains a filter, a resin, which basically at the end of this purification, the system is going to take the eluent and it's going to dispense it into that filter bed. And that filter bed is going to be removed by the scientist, capped and centrifuged offline. That final centrifugation step is going to get you to that really pure plasmid by basically removing any potentially carryover of magnetic beads, as well as any potentially leftover endotoxins in the sample. This is a key step to ensuring you're going to get a very good, clean plasmid. Now I'm going to be showing the user interface for the AmMagQuattro. What you're seeing here is basically the interface with only one module connected. So you had seen earlier in my slides that basically the controller is capable of controlling up to 4 modules. Keep in mind, you do not have to have a 4-module setup, of course. What you're seeing here is actually a single module setup. So this is where module number 1 is basically online. The second overview I want to show is really essential in ensuring that you're set up correctly. I think most people that have worked with automation would agree that typically any error that ends up taking place onto the automated workflow is typically because of the setup, which was done either incorrectly or maybe lack of training, or potentially something was not set in the right place. So what you're seeing here is one of the key steps of ensuring the user's success. So part of our development, we implemented a scanning feature into the AmMagQuattro. So once you have your deck set up, the instrument is going to go ahead and prompt you to scan the deck, and it's going to indicate whether or not the instrument is set up correctly. So it will give you a visual indicator of what is on the deck. So you can see there's a tip with the 6th position, there's a reagent, there's a reaction tube, a sample tube, as well as the waste container. So in the scenario that you did not fill up the instrument up to 6 samples, it's not a problem. You can certainly not fill that full capacity, the 6 samples. You can do 2, 3, whatever it is that you want. But the system will also give you a visual indication here, showing you that those positions are empty. So again, this is a key attribute to ensuring success. Next thing that I want to highlight here is the capability of the AmMagQuattro basically providing you with a real time of what is happening onto the instrument. This is actually one of my favorite things about it, because in a lot of cases, what ends up happening is that obviously the scientist sets up the deck, they hit start, they potentially get an estimate of how long the run is going to take. But in a lot of cases, they get multiple different projects, they get distracted, they don't know how much time is left on the run. What's really nice about this feature is that you can walk up to the instrument at any time during the run. This setup is going to give you a visual indication of what has been complete and what has yet to be complete, as well as the status of the as well as the countdown of how much time is left over on this particular run. So for this example, you can see that we have done the resuspension lysis neutralization, and we are currently at the pre-clarification step. We have yet to do the remainder of the steps that are grayed out, and we have 35 minutes and 23 seconds left on this run. So a very detailed overview as well as a countdown of how much longer it is left to be ran. So now I'm going to be showing you performance data specific to the AmMagQuattro. I'm really excited to show this because it really speaks to the quality of what is being generated on the AmMagQuattro. And in addition to that, it actually also contains data from clients. So we've had this solution for a couple, I would say it's been about a year and a half now, and it has been a huge success. So you can see that the data I'm sharing today is, of course, internally generated data, but also data that is being generated hand in hand with our clients. So the very first case study that you're seeing is to compare the AmMagQuattro's performance next to basically market available kits. So in this particular scenario, we ran 2 different plasmids and we eluted with different volumes. Overall, you can see that the AmMagQuattro concentration was about 1,000 nanograms per microliter, and the manual concentration was about 1,500 nanograms per microliter. However, the elution was lower for the manual. It was one of the first internal case studies. I still like to show it because it is one of the first steps that we did. But you can see that basically the quality, the OD readings, as well as the endotoxin levels and the supercold ratio of the end product was high quality, and it was consistent with what we saw with the manual kit. The next case study compares different culture media. So whenever you're doing plasmid growth, there's going to be quite a bit of different factors that are going to impact the outcome of your bacterial success. So if you're working with plasmids that are high copy versus low copy, if you're working with a toxic insert, if you're working with a very rich media, such as a terrific broth versus LB, or maybe a different media, the outcome of your experiment is going to vary very, very heavily. So what you're seeing here is that we basically grew the same bacterial culture in LB 100 ml, and we also grew that same bacterial plasmid with TB. So you can see when we weighed that bacterial pellet after centrifugation, we got an average of about 0.8 grams for the LB. On the other hand side, the TB was about 1.2 grams. We ran those side by side, so that was really the only thing that was different. And what you're seeing here is that the concentration for the finally eluted DNA was almost double for the LB versus the TB. So with that said, sorry, I think I have an error on my slide. Sorry about that. With that said, basically, the TB prepped sample was highly more robust, basically attributing to quite a bit of DNA in the TB. Another case study I wanted to show was actually a case study that we did with a client. What we did here is basically use only 30 ml. That 30 ml was sufficient to get us to that 1.5 gram of bacterial pellet, and we eluted the sample with about 70 UL. The concentration of the sample was approximately 1,000 nanograms per microliter. The next case study I wanted to show is a side-by-side comparison for basically running the standard protocols versus running a protocol that was further optimized. We have a really wonderful support team, a field application scientist team that are going to work with you if you end up obviously choosing to work with the solution. And what you're seeing here is that when we ran a standard protocol, we got out of that particular protocol approximately basically 500 nanograms to 600 nanograms. After optimizing that protocol, we got close to 1 mg of plasmid DNA, which is being shown on the right, the column from the second last, showing the yield. The next case study I wanted to share is also a case study that was done using with a customer. So what you're seeing here is that the client basically took the AmMagQuattro, they purified their plasmid on it, and they took those 3 samples and purified those exact same samples on using a manual kit. Overall, the concentration was slightly higher for the manual kit, but the quality of the actual plasmid was fairly comparable. After that, the client actually did a digest to check the actual plasmids, and you can see that there was no observable genomic DNA, no observable RNA, and the preps basically looked identical for the manual versus the AmMagQuattro. After that, the client took those plasmids, both the 3 from the manual prep as well as the 3 from the Quattro, which is the automated prep, and they did a average or a transient transfection. And they basically tested the average titer of the protein. So you can see overall the exact same plasmids, even though there was a slightly more for the manual prep, a higher concentration for the manual prep, the transfection was spot on. There was really not much difference. I would say for plasmid number 3, the Quattro showed a little bit better, but overall it was very comparable to the manual kit, which of course speaks to the quality of the plasmid. Next thing that the client did was also take that same plasmid and use a transient transfection system that they had automated. So here you're seeing basically the plasmid that was purified on a manual setting versus the plasmid that was purified on the AmMagQuattro. However, you're seeing it being transfected on an automated system. So you can see here once again that there was no real observable difference between the AmMagQuattro plasmid versus the manual prep plasmid. I would say really the only one that showed somewhat of a difference would be plasmid number 3, where again the AmMagQuattro achieved better results on the lower right-hand side. And this is one of the last case studies I have to show. Basically what we did here is we took one of our services plasmid, which was prepared using a manual kit. I think a lot of people here must be familiar with the GenScript services division. They're really well known for high quality plasmids. They actually provide that as a service. So with that being generated by our services division, the idea here is that we're having basically a third-party plasmid coming into our team and basically as a positive control. So what you're seeing here, the plot that is in blue, we did a luciferase reporter gene. We're looking at basically the fluorescence of the, to test whether or not the transfection was comparable or as efficient or potentially even lower. So by doing that, we looked at the manual prepared plasmid. We did 2 sets of plasmids using the AmMagQuattro, which is the plot in green, as well as the plot in purple. And the last one was basically a negative control, which was just the HEC 293 cells. So what you're seeing here is that the AmMagQuattro plasmids, the plot in green as well as the plot in purple, actually outperformed the manual prepared plasmid. And the last case study I have is specific to a P-lentiviral titer. So what we did here is, once again, we took the AmMagQuattro and we purified a P-lenti vector on it. And then we took that same P-lenti vector and we purified it using a manual kit. The table on the top, you'll see that we basically purposefully did an elution of a 1.5 ml using the AmMagQuattro, while the manual kit, we did a 500 UL elution. So basically a 3-fold dilution for the AmMagQuattro. And once again, we did that on purpose, and you'll see why in a minute. But the concentration for the AmMagQuattro ended up being close to 300 nanograms per microliter, and then the manual kit was about 1 mg per ml, which of course is not surprising because it was 3 times more dilute. So after that, we took basically those two plasmids that were purified in the different methodologies, and we did a P-lenti transfection. And after that, we did the testing of the average titer. And you'll see on the right-hand corner at the bottom of the slide, the average titer for the AmMagQuattro for the two first samples was about 53 or 49. Average titer, and then for the manual was about 50 or 52 once again for the average titer. So you can see here, even though the AmMagQuattro plasmid was a 3-fold dilution, so significantly less concentrated, even with that, the average titer was still very much comparable to the manually purified plasmid. With that said, the point here that I'm trying to say is that the quality of the plasmid, even though it is less concentrated, it is not being jeopardized. So it's a really robust purification method, enabling you to get really the best transfection efficiency for your sample. We have quite a bit of happy clients globally. And I wanted to share with you one particular quote from one of our happy customers. This is Dr. Zhu from T-Scan Therapeutics. He is the head of the vector development and cell engineering therapy at T-Scan. He basically mentioned how much how happy they are with the instrument. He said that it's really has exceeded their expectations. They basically have been able to save quite a bit of time on not just time, but also cost. And Dr. Zhu is actually on our website if you'd like to read the interview we conducted with him. Now I'm going to be showing you a video of the AmMagQuattro, but before I do so, I did want to highlight a couple things. The instrument itself, as I mentioned earlier, is designed to be coupled with the consumables that are being provided as part of the solution. So you'll see that the actual cartridge is sealed when we load it onto the instrument. That sealed cartridge is going to be containing all of the buffers, all of the magnetic beads, all of the actual elutions and washes. So that particular cartridge, the instrument is actually equipped to be handling that particular cartridge. Another key feature is the in-deck spinning and magnetic modules that are available in the deck. So you'll see that the AmMagQuattro is basically going to be spinning the samples as we're pipetting in and out of them. This is going to enable us to really wash the samples as well as get a really good bind on those magnetic beads. And you'll see basically those steps I had mentioned in a minute. All right, so now I'm going to be very briefly touching base on our newest advancements. We're going to be enhancing our offering. We're working on a couple different projects right now. I'm just going to give you a quick sneak peek into that. So one of the big things is really optimizing our current solution. So we're working right now on making that optimal range of bacterial pellet that I had mentioned earlier in the presentation, the 1 to 1.5 grams of bacterial pellet. We're trying to expand that and obviously handle a larger amount of DNA. We're also looking into enhancements or expansions into additional applications. So we'll look forward to launching that soon. I'm not going to go into a lot of details about that. And the last thing I want to highlight is that we all are also working on accompanying solutions. So we are also investigating and developing, basically making additional automated solutions that also can go hand in hand in this. And with that, I'm going to be talking about our automated as well as manual antibody and protein purification solutions. So some people might be very familiar with this instrument. We've had this solution for a couple years now, what we call the AmMagTM. This is an automated solution for antibody and protein purification. What's really nice about it is that it takes a tedious workflow from, multiple hours to less than an hour. And the nice thing about it is that it can handle up to 12 samples in 1 hour. So significant reduction in not only the labor, but also the amount of time that you're going to get that purified sample out of it. A really nice thing about it as well is that it is magnetic bead based. So you're not going to have to basically do any centrifugation. You're not going to have to basically pack columns. You're going to go ahead and take your beads. You can even incubate it with your culture. Take those cultures, load them onto the instrument, and then basically hit go, and you can basically come back and get your elution. Another nice thing is that the elution can go as low as 40 UL, and we do have about, I want to say seven different solutions that you can use onto the system. A really nice thing as well about it is that we have set it up where you can do your own protocols, and you can define, of course, the volumes, you can define the number of washes, the conditions of the washes, multiple pipetting, things like that, as well as the number of elutions. So you can definitely do like a sequential elution. And with that said, I have a quick overview of showing the performance of the AmMagAssay. What you're seeing here is our protein A magnetic beads. I do want to mention that we have a portfolio for magnetic beads that go with this solution. So the magnetic beads are up to you how, of course, most of them are affinity based. And what you're seeing here is a resin system that we're comparing it to. So we have approximately 20 plus samples here. And what we did here is side by side, we purified using the AmMagAssay protein A beads and the resin system. Another table that you're seeing here, another plot that you're seeing here is the binding capacity of the magnetic beads. So on the right-hand side corner, you're seeing basically the competition, which is the yellow bar versus the GenScript protein A beads. So just a quick overview of the capacity of how much protein we can bind. Very briefly here, I wanted to mention we do have quite a bit of magnetic beads, as I mentioned. Some of them could be reused, which end up being a really nice thing for academic users, especially because you get huge cost savings. You go from a few dollars to a few cents per sample, so significant cost savings. Those same magnetic beads can technically be used using the automated racks, using the manual racks, so it doesn't have to be on an automated fashion. And as I mentioned before, we have a couple different offerings for both antibody as well as tagged protein purification. So we have protein A, protein G, we have a couple different nickel charged, and of course the FLAG tag. In addition to that, we have a couple different enzymes, such as an endonuclease, what we call the benzneburase, that ends up really reducing the sample viscosity and making your sample a lot more manageable. So of course, any contaminating cell debris, any contaminating nucleic acid is going to make your sample extremely viscous, which makes it challenging to handle. We have also quite a bit of different precision tag cleavage enzymes if you choose to use that at the end of your purification. And then we have a nice portfolio for both endotoxin detection and removal. And the last thing I want to highlight here is we also have a wide portfolio for our protein purification resins. If you're already basically using resins and you really don't want to switch, but you are looking for a more cost-effective solution or a better solution than what you currently have, we actually have a lot of clients that say that our resins are really one of the best performing in the industry. We have a lot of nice solutions for that, especially when we look at the immunoprecipitation. I'm going to skip the slide for the sake of time, but I did also want to switch gear and talk about our protein electrophoresis and Western solutions. I think that probably quite a bit of work is being really done to this day using the gold standard for immunodetection, which is, of course, Western blotting. So, of course, even though this is a very good technique, it is still fairly labor intensive and is extremely time inefficient. It takes multiple days. It also it's riddled with inaccuracies and inconsistencies, especially if you're going from one user to another. So the reproducibility is fairly challenging. So one of the first solutions I want to present before I dive into all of it is really the gels that we are offering. So this is a key component, of course, in any Western workflow. So we do have bistress gels. These gels are really nice in the sense that not only are we offering multiple different concentrations and different gradients, we also are offering a larger wall volume, so it can handle up to 80 ULs, which is not something most market offerings have. Another thing to mention here is that because of the formulation of the bistress, it is actually more of a neutral pH, which minimizes the polyacrylamide hydrolysis, which of course does a lot of stability, so a lot less impact on your protein. So with that, you can see quite a bit of data that we have generated. I'm not going to go into that because of the sake of time, but I did want to mention before I go into the next slide that we have taken the Western workflow and provided a complete offering for every single step of the workflow with multiple different devices. The first device I'm going to go into is the eBlot, which is a fast wet transfer system. So I think a lot of people are probably familiar with the gold standard for transfer, which is really the traditional wet transfer using manual methodology. There are a couple instruments out there that automate that process. However, none of them are wet transfer system. So they are either dry or completely dry or semi-dry. And while that definitely reduces your time, it is still fairly limited as the results end up being inconsistent, as well as the transfer is not going to be efficient. So what we have is actually the eBlot L2. As I mentioned before, it is really the only wet transfer system in the market. And what we have developed is what you're seeing on the right-hand side is a very unique and clean setup for the transfer sandwich. The second slide I have for you is to show side-by-side comparison all the way from a 7 kilodalton protein to 170 kilodalton protein. So you can see, because of the fact that we are using a wet transfer setup, there's really no limitation for the protein size. You can go all the way from a 3 kilodalton to a 1200 kilodalton. For this particular example, 12,000 kilodalton, I'm sorry. For this particular example as well, on the right-hand side, this was actually a publication from one of our clients, and you can see that they did a 12,000 kilodalton. Next slide I want to show is the Easy West Light. This system actually completely automates the Western blotting workflow for you. It is equipped with a really nice user interface, and it has a removable cassette where you can basically load your antibody into the cassette with your gel, and you can take that and basically incubate it at a 4 degrees C. What's also really nice about it is that the reagents are recoverable, so you can remove your antibodies that were not used and basically recover them. So minimal waste. I also want to mention that we recently launched our Easy Diluent Kit, and you can see that the results for the Easy Diluent Kit is really optimal because obviously it reduces significantly the time. as well as, of course, with the Easy West, you're completely automating the workflow. So hand in hand, those two solutions end up really providing you with not only significantly shorter time to run your samples, but of course, obviously optimized results. And this is where you're seeing a quick overview of the different samples, beta-actin, A-tubulin, and HSP sample. And then the last device I have to share is the eStain. This is an automated Kumasi blue stain. As I mentioned before, this is kind of completing the workflow of the Western workflow. What's really nice about it is that it has really quick workflow. We can do a 10-minute stain and destain for 2 gels simultaneously. It's also very nice. So it's taking a super messy process and automating it for you. Instead of doing multiple hours, you're basically automating it in a few minutes. And you're not having to deal with a lot of liquid and a lot of that messy workflow. We also have seen a lot of success cases because of the high sensitivity of this instrument. So it can actually detect about 12.5 nanograms. So ultra sensitivity here. And the next thing I want to mention here is that it is also compatible with the Ponzo S solution. So if you want to do membrane staining, that's certainly an option for this instrument as well. And then the last thing I would like to highlight is basically with this complete workflow, you're going from approximately a 2-day traditional workflow from electrophoresis to transfer to your Western to approximately 2 hours with this workflow. So it's a really nice setup. It's completely automated. It saves you an immense amount of time as well as labor, and it has consistent quality. And with that, I'm going to be talking very briefly about our cell isolation and activation solution. So I think a lot of people are very familiar, as I mentioned earlier, with an antibody development platform that we have. GenScript is really well known for that. We've had it for more than 20 years now. And what we do is basically have a very robust platform to optimize antibody discovery. So we took that discipline as well as our 10 plus years of magnetic bead development, and we combined those two business units to enable us to basically offer solutions for our cell and gene therapy market. This is somewhat still newer for our portfolio, so we are still looking for partnerships. And I did want to mention as well that this was also in conjunction with our Legend Biotech division. This is a cell and gene therapy company. They're really well known. So with that said, if you are interested in partnering with us, I am actually looking for people that would be interested in perhaps publishing with us or maybe beta testing with us some of these products. If you are interested and you are working specifically with cell therapy, cell isolation, cell activation doesn't have to be cell therapy. But if you are working in that space, feel free to reach out to me. I'll leave my information, of course, at the end of this presentation. With that said, I'll go into the actual solution. So we have what we call the CytoSync platform. Specifically, we have magnetic beads that are nano-sized, about 100 nanometers. They're extremely stable, and what's really nice about them is that they're actually biodegradable. So the additional steps that are typically done to remove any kind of the magnetic bead setup when you're working with cells is really not necessary here because they are biodegradable. Another very important factor here is that they're extremely cost efficient with excellent performance. I would say we have one of the top performances in the market. Historically, there's only been 1 or 2 vendors in the market, especially for the smaller size of magnetic beads. So having an additional offering, an additional vendor that you can rely on is certainly something that I think a lot of people would benefit from. I also want to mention that we have both research grade as well as GMP grade offerings, and we are constantly expanding our offerings. So right now we have about 10 different products, CD4, CD8, CD3. I'm not going to go into each one, but we are constantly expanding those products as well. So if there's something you don't see and you would like us to maybe look into it, once again, I want to encourage you to reach out to me. So overall, very briefly, I did want to mention that basically we do have obviously both the manual workflow as well as the automated workflow for cell isolation. And I did want to briefly touch base on our Cytosync 1000. It is completely automated, large scale, high throughput. And this is some data that we showed side by side using CD4, CD8, basically side by side using our instrument. And with that, very briefly showing you the overview of the different product portfolio for cell isolation. You can see we have the reagents, including both isolation and activation, the consumables such as the columns, as well as the tubing sets for the instrument, and lastly, the accessories or the racks, as well as the automated system. And with that, I wanted to kind of leave you with this overview slide. We have solutions all the way from kits to assemble the DNA constructs to the automated platform that's going to enable you to purify maxi-scale plasmids, as well as an automated platform and a manual platform for antibody and protein purification, as well as the accompanying consumables and reagents that go with it. A full protein workflow and Western blotting, as well as the cell isolation and activation. We also have quite a bit of different products, but of course, I'm not going to go into every single one. With that, I kind of just wanted to leave you with the fact that we have a full support, not just with my division, but with multiple other divisions in GenScript. And I thank you for your time, and I'm happy to take any questions now.
Well, thank you so much, Rouba, for your informative presentation. We will now start the live Q&A portion of the webinar. So to our audience, if you have a question that you'd like to ask, please do so now. Just click on the Ask a Question box located at the bottom of your screen. All right, now let's get started. Our first question asks, do you offer demos for the instruments you are showing? And what about academic discounts?
Yes, we certainly do. We actually have a demo program. I'm happy to answer that offline or right now for the purpose of time. But so far, we've had a lot of success with all the solutions that I have shared. So a lot of our demo is a little backlogged. So with that said, I highly encourage you to reach out to your account manager or to me, and we can kind of figure out what would be the best timeline for you as well for us. So for example, if you're really rushed and you want to evaluate it fairly quickly, we might explore the demo to buy program. However, if you're okay with waiting a little bit, I think a better program would be a virtual demo where we schedule a call for you, you send us your samples, and we actually run your sample with you online, and then we ship it back to you for additional analysis. So either option is certainly available. Of course, the virtual demo is going to be extremely cost effective, not just for GenScript, but also in the sense of the complexity of the workflow as well.
Great, thank you. And we're running short on time here, but we've got time for one more question, so we'll wrap up after this one. Now, how can I integrate GenScript's Western blotting solutions into my current workflow?
That's a great question. Thank you for that. I think that really one of the main things about our instruments is that it's really taking the tedious steps out of your process and automating them for you. So it includes electrophoresis, this transfer, the staining. It is all being done basically using these solutions that I had presented. So whether you want to do the whole thing with us or you want to, there's just one step that's extremely tedious for you, you can certainly start with that and see how that goes. But of course, it kind of also enables you to focus your effort on other things that might be more beneficial for your research. Another thing I want to mention is that the actual size of the instrument is very compact. So even if you don't have a lot of bench space, which of course is a big pain point in automation, you're not necessarily limited with the space.
Great. Well, thank you again, Rouba Najjar, for your time today and your important research. Be sure to also check out GenScript's booth located in the exhibit hall for more great information. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. I'd also like to thank our sponsor, GenScript, for sponsoring today's webinar. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 1 year until March 20th, 2025. Labbers will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.